Hello everyone, this is Agri of Videos and the topic of today's video is seed and we are going to talk about the different types of seed. In this video, we are going to talk about the monocot seed and the dicot seed, what is the difference between the two seeds and the different seed tags which are used in a few organizations and associations. So let's begin. First of all, what is a seed? If we name it in form of a definition, it is structurally true seed is a fertilized mature ovule. It consists of majorly three parts. One is called the endosperm, second is the embryo, and the third is the seed coat. This endosperm is called as the storehouse of the food. Seed coat, this is a protective layer, and embryo is the very important part of the seed. Store of the food, if we say, it consists of two main parts. One is the endosperm, and the another one are the cotyledons. So, what is a cotyledon now? Because in the subsequent slide, we'll be talking much in detail about the cotyledons and the phenomena of being a monocotyledon or a dicotyledon. So, this is a part of embryo within the seed of the plant. So, it will be located in the seed of the plant. And what is it defined as? It is the embryonic leaf in the seed bearing plants, which first appears from a germinating seed. So, first emergence happens of the cotyledon. So, this is the embryonic leaf in the seed bearing plants. And what is this endosperm? As we have already talked about, this is acting as a food storage house for the developing plant embryo, which consists of starch with proteins and the different nutrients in that. Now, talking about the two type of cotyledons, which is monocotyledon and the dicotyledon, or we call that monocots or dicots. First line is absolutely clear. Single cot and dicot has two cotyledons. Monocots will usually have this endosperm. They are endospermic. They have this endosperm. But dicots, they usually are non-endospermic. For example, if we are saying exceptionally, we have exceptional cases are there. An example is castor seed in that. Okay. Germination in case of monocot, it is hypogeal, except in case of onion. And in case of dicot, it is epigeal. What does this mean? Epigeal germination is when the cotyledons emerge out of the seed. And in hypogeal germinations, the cotyledon will remain inside the soil. If we are going to compare them, then in the monocotyledon, the this if we say here, the cotyledon will remain inside the soil. This part is inside the soil. And in this case, if we see, the cotyledon is above the soil. You can see here clearly, this is located above the soil. Then flower parts in multiples of 3 are present in case of a monocot. And in dicots, the flower parts are majorly in the multiples of 4 or 5. Major leaf veins, the leaf vein in case of monocot, it is parallel. And in case of a dicot leaf, it is reticulate in shape. Okay. Stem vascular bundles, they are scattered in case of a monocot. And the stem vascular bundle is in a form of a ring in case of a dicot. Roots, if we talk about, then in case of a monocot, the roots are adventitious. And in case of a dicot, the roots are developed from the radicle. Secondary growth in case of monocot is absent, but in dicots it is prevalent. If we talk about examples, then we have grains like wheat, corn, millet, rice, sugarcane, banana, ginger, onion, grass. These are all examples of monocotyledon seeds or the monocotyledon plants. Dicots, if we talk about, then legumes, mint, lettuce, tomato, oak tree are all examples of the dicots. Now there was a term radical. What this is radical? What is this radical? This is the first part of a seedling. Like you can see in here, this radicle. What is this? This is the first part of seedling growing plant embryo which emerges from the seed during the process of germination. So this is coming epicotyle, this is hypocotyle, this is endosperm in this case, cotyledon, coleoptile, epicotyle, hypocotyle in this form is there, and then we have the seed coat, then we have radicle in this case, radicle here. You can see it very clearly. Now, if we are talking in detail of each one of them. So, fibrous root in case of a monocot and tap root system in case of a dicot. First thing. Okay. Monocots, they have one cotyledon, two cotyledon, everything is done. Then veins also we have talked about parallel veins and veins like a net or reticulate. Then we had talked about the vascular bundles. Monocots have structure like this. They have scattered vascular bundles. In case of a dicot, the vascular bundles are very properly arranged in form of a ring. Flowers also we have talked about that in the multiple of three, and in case of a dicot, multiple of four or five. This is also clear. Root system is also clear. Everything is done here. Okay. Now, when you are making the picture of a monocot or a dicot seed, so monocot seed is going to look like this. It is having the endosperm here like prevalent. And in case of a dicot bean seed, you can see the structure of a seed and the presence of endosperm is here. First true leaf, seed coat, and the cotyledons. Here, monocot, seed coat, single cotyledon, leaf sheath is present. First true leaf, embryonic root, this is all prevalent. Now, once the seed is sown into the ground, germination happens and the root system develops. This we have talked about, parallel veins and the net-like veins or the reticulate veins, all we have done. Leaf sheath is prevalent in this case, not can seen in the dicot. This is how it goes. Now, if you're going to detail about it, just remember the things we have picked from there. In case of a monocot, you can see the endosperm. You cannot see it here. Seed coat is prevalent in both of them. We have epicotyle, hypocotyle. Epicotyle is prevalent here. Cotyledons are two. Cotyledon is one. Radicle is prevalent here. Example is bean. Monocot example is corn. Now, seed undergoes few production stages. What are those stages? Once the seed is sown, you are only able to see after it is planted or it is after the production stage. So here, if we are going to talk about the different stages, also I have talked about this the same in a different video. You can refer that. I'll mark the link here. Initially, first is the nucleus seed, the basic seed. Second is the breeder seed developed by the breeders under various supervision activities. Then we have the foundation seed, followed by that is the certified seed, and finally the seed is going to go on the farmer's field. So now is the time when farmer is receiving the seed. So you need to know the sequence: nuclear seed, breeder seed, foundation seed, certified seed. This is how the sequence goes. Okay. These are the different type of seed. These are the agencies involved. Original. I have already talked about the same in a different video. You can refer that, or you can just have a snip of this here. Nuclear seed agency involved is the original breeder. In case of a breeder seed, it is a sponsored breeder, ICAR, Institute, National Seed Corporation, and state federations for the same. Foundation seeds are majorly developed in recognition with the various seed producing agencies. Certified seeds, it is all governed by the private seed companies, NSC, state, and the different partnerships. Nuclear seed has no tag color. Breeder seed is generally golden in color. Foundation seed is white in color. Certified seed has a color called azure blue. The different sizes of the tags are also mentioned here. If we talk about the purity, the nuclear seed is genetically and physically 100% pure. Same is the case of with the breeder. Foundation has a purity of 99.5%, and the purity in case of certified is restricted to 98% approximately. This is distributed to the farmers in the commercial cultivation. Now, what is genetic purity? You should know if you're talking about seed, genetic purity and physical purity. What is the true definition of them? We should talk about that. So, genetic purity, it is the trueness to the parent type. How true that seed is identical to the parent type. That is called as a genetic purity. And what is physical purity? When you are physically able to visualize and see the difference, like if there is any inert matter present apart from the matter which is the seed. If seed is of this shape and you're finding a subsequent thing which is not identical to the seed, you're able to visually check it, see it. That is where it is called as the difference in the physical purity. There is when broken seed is present, in case we have different weed seed present, soil, dust particles, etc., shaft, different things are there, and then thus the purity gets disrupted or purity gets compromised in that case. So I hope this is clear. Now moving forward, we have few organizations that you should know. You should be reading about this now. National Seed Research and Training Centers, which is a center which is located in Varanasi. Then we have CSFCI, which is State Farms Corporation of India Limited, which is responsible for the quality seeds availability on the various state level grounds. Then we have National Seed Corporation Limited, which is responsible for the seed production and certifications in case of certified seeds and foundation class of seeds. Cooperatives are also involved in case of seed like CRIPCO, NCDC, then we have NHRDF, then we have FCO, then we have NAFED. There are many, many organizations like this. You can refer to them, each one of them individually. Then there is an International Seed Testing Association, which is also called as ISTA or ISTA. The objective of this association is to develop, to adopt, and to publish the different standard procedures for the sampling, for the testing of the seeds to make a uniformity across, and the process of evaluation comes across easier in the international trade, on the terms of trade when we talk about. Second is to promote the research in all the different areas of seed sciences, technology, including the sampling, testing, storing, processing, distributing, and many other activities which are involved in the process of seed formation. So, this is all about the chapter. I hope you liked the content. Thank you so much for watching. Do give it a like. If you are liking the video, please subscribe to this channel. If you have any comments, please do mention it in the comment box below. Thank you so much for watching.